analysis workflow. And so I just want to take a few minutes. I added some new slides to the slide deck this morning just to sort of orient everybody and hopefully get us on the same page there. And then I just want to mention at the end, we're going to give another questionnaire today. Please do give us any of that kind of feedback, because as you see, we're adjusting what we're doing here based on what we hear is working, what we hear is not working. So after we do that, after we reorient a little bit, I'm going to turn it over to Chloe, and Chloe is going to go through several QIIME2 analyses that are pretty standard, like almost every workflow, every analysis we do, these are things that we integrate in that workflow. And then we're also going to show, just to sort of relate Docker and Galaxy to each other, we're going to run a couple of those steps that we ran yesterday afternoon in our Docker containers. We need to get that same data, need to get it loaded up. And so we're going to spend just a few minutes on that too. And then at the end, we're going to wrap up with the questionnaire again for a few minutes. Okay, so first thing is, if you could just navigate back to this written tutorial that we have. And as a reminder, if you were to go to our slide deck, so to this QIIME2 slide deck, this would be that tutorial link at the bottom of the page. And so then when you get there, click on the left side on the working with your QIIME2 container. This document should be familiar. This is what we gave you for homework last night to get through the first couple steps in here. And so last night, maybe you went a little bit further, but at the very least, what we wanted you to do last night was to basically run this command in the Docker terminal. You got a little bit of more context today on what that was doing. And so what this was doing is it was going out and it was grabbing this QIIME2 container, pulling it down to your computer so that you could launch, pulling this image down to your computer so that you could launch a container that's going to have QIIME2 and various other tools installed in it. And so the next thing that I want you to do, if you haven't done this already, is go ahead and copy this command. There's two tabs here, one if you're doing it with Podman, one if you're doing it with Docker. Select the one that's relevant for you and paste that into your terminal. So just similar to what we were doing this morning. And this is going to create this data volume that you'll be storing data in. So go ahead and do that. If you already did that and try and do it again, it's probably going to complain that you already have that volume. Nothing to worry about in that case. And then what we're going to do is start that container so that we can then get in there and start working with it. And this is a container that we're going to interact with, not through the terminal directly, but Candace mentioned, sometimes there's these containers that have things like Jupyter servers running in them. This is an example of that. So go ahead. I have, so I'm working on an M series Apple up here, and I'm using Podman. And so I'm just selecting the tab that's relevant for me. You pick whichever one is relevant for you. So I'm going to copy that. If you, navigate to like the top right of these boxes, there's a little copy button that shows up. And when you copy it, it'll check it off and let you know that you did that. And then I am switching back to my terminal window here. I was doing a few things just before we started. And so I am just simply pasting that command in that I copied. I'm going to hit enter. This is going to run for a second, and then you should see that you get a command prompt back. And so really, it looks like nothing happened here. But what actually happened is that it started up this container in the background on my machine. And so if I go back to my instructions here, you can see that I have this interact with Jupyter Lab and QIIME2. So when the container is running, you can interact with it by opening this link. And so you should have this link. If you click that, you should see this Jupyter Lab environment start up. So I'll give everybody a few seconds to do that. If you get stuck, if something's not working, if you don't see what I'm seeing up here, raise your hand and somebody will come around and give you a hand. All right, we've got a couple hands going up. One more. So I see two folks over here. Did anybody else need help? Okay, so that means everybody should see what I'm seeing up here. Okay, so this is now, I'm just zooming in, so it'll look a little bit different for me. I just zoomed in a couple clicks. Now, this is, like Candace was saying this morning, this is this environment that's now standardized across all of our computers. So whether you're running Mac OS, Windows, or Linux, this should all look the same, and the way that we interact with this is going to be the same. And so what you should do here is click this terminal box. And so that's the one with the $ symbol in it. And this will take just a few seconds this first time, maybe up to 10 or 15 seconds. But what this is doing is the computer right now is just compiling some information about the installation of QIIME that's in here so that it can run more quickly. When it's done, you should see a prompt that looks like this. And so just to make this like a little bit more intuitive for folks who don't frequently work in this environment, we just kind of made this fancy command prompt here where it tells you what your current working directory is. And so in this case, it's showing, it's a little hard to see on my screen, but it's a tilde. And that is just sort of a shorthand for like your home directory. Like this is where you're always starting. And then the $ is just our command prompt. And so that's just telling us that the terminal is waiting for us to type something to give it a command. And so let's type our first QIIME command in here. And the command we're going to type is just QIIME -- or QIIME space -- help. And so when you do that, you'll see some information that comes back. I just have to scroll on here because I'm zoomed in. But basically, this is telling you you are working with the QIIME2 command line interface. It's giving you some information on like different options and then the different commands that are installed in this environment. And the commands are different plugins that are available in this environment that we're working with. And so a lot of these are, if you've worked with QIIME2, a lot of these are ones that you're familiar with. Hang on just one second. Yeah. Yes, it's localhost. So there's a link in the book. And so it's localhost :8889. Got it. Okay. So I'll just talk through this while a few people are still working on it. But so basically, this is like the list of QIIME2 plugins that are installed in this environment. A lot of these are probably familiar. And so like metadata is one that we were interacting with yesterday. And so let's just go ahead and get some information on that metadata plugin. So what you would do there is you would type QIIME metadata -- help. So QIIME space metadata space -- help. So I'm going to run that. And you can see that this comes back with a list of subcommands that are available through this plugin. And so this is kind of like how we think about organizing functionality that's accessible through QIIME2. And so if you look like at that last one that shows up in the list, that's tabulate. And that's what we were working with briefly yesterday afternoon through Galaxy. And so if you know, like this is a command that you want to work with or you're exploring commands, you could then check out how that one might work. Probably guessed it. QIIME metadata tabulate dash dash help. And what this is then going to do is describe what are the inputs, what are the outputs, what is the functionality that's available through this particular action that you could take with QIIME2. And so up here, when we look at the help text for this, this says, generate a tabular view of metadata. This output supports interactive filtering, sorting, exporting. And so this is basically what we did yesterday. Okay, so that's like our basic QIIME2 environment. The other thing that's kind of neat about this interface is we have this file browser over here. And so what I want everybody to do is we're going to create a new folder or a new directory that we're going to work in. And so the command that we use for that, and I have this documented in another page in that Jupyter book. Let me just show you that real quick. So if you navigate to this next one, exploring the data through your container, I'm going to walk you through this, but this gives you some information. These are the commands that I'm basically going to be working with right here. And so I'll do this up here, and you can refer back to that document. But basically what I'm going to do is I'm going to make a directory called 2024-workshop. And so I'm using the command mkdir for that. Mkdir stands for make directory. I'm going to hit enter. And that will run and come back. And I was expecting it to pop up over here. Yeah, it just took a second. But what you can see is like this is our essentially like our Finder window or our Explorer window over here on the left. And so this 2024-workshop, that's that folder that we just created. We could also, create a new folder over here on the left. We could upload files over here on the left. And so if we had some data files on our computer and we wanted to get them in here to work with them, we could just use this little upload button. And then this refresh is just, if it's lagging like it was for me for a second, that'll just refresh that browser. Another way of getting the same view would be to use the tree command. This is slightly different from how we have it in the book. But if I run this tree command, what that's doing is it's basically showing me the same thing that I have over here on the left. You can see there's this 2024 workshop directory. We have this workshop input data directory. And that's just kind of like our backup. We're going to have you download these files directly. But if for whatever reason we didn't have an internet connection in this room right now, we would still be able to continue on with this event. And so this is the environment that we're going to be working with for QIIME2. And then this is an environment that you could take home with you. You know, this is running on your computer right now. And so this is like perfectly good environment to really start like your learning of QIIME2 as you go home. And so all of our tutorials that you can find on our website would work directly in this environment. You don't have to install anything extra. And so you could just dive right in with this. And then one of the things that's very neat about this is that you could, when you're ready to start moving on and doing some analysis of bigger data sets, there are ways that you can use this same container, but make use of cloud computing resources. And so like everything that you're doing right here, you could then just scale up to bigger computing resources. And so it still look and feel the same to you as the user, but on the back end, it might be running on a cloud service. If that's something that's of interest to folks, feel free. I don't want to take a bunch of time talking about that because everybody's needs are going to be a little bit different. But we're here. I'm more than happy to chat about that with you. Colin has a lot of experience with configuring those kind of environments. And so he would be a great person for you to chat with about that also. Okay, so I'm going to jump over to my slides, and just for a few minutes, I want to address that feedback that we got yesterday. And Kate had me pulled up on the right slide here, but then I messed with everything. And is that it? Yeah. Okay. So I just want to really briefly work through what some of the files are that we've dealt with already, and just kind of contextualize that for what we're going to be doing in the next stages. I realize a lot of this, people have some experience with, know about this already, but I just want to use this as an opportunity to get our language consistent, get on the same page. And so generally a microbiome study is going to start with going out into the world and collecting some samples. For our purposes, we don't really concern ourselves too much with what those samples are, but maybe these are like fecal swab samples. And so you might collect them with a sterile swab like the one that I have up here. And you may have some sample identifier associated with each one of these. And so in this case, I've got this sample identifier for a C2, and then that is going to relate to some information that I have in a sample metadata file that you would build for use with QIIME2. Now, sample metadata files, I will point you at some documentation on how you generate those. But QIIME2 really doesn't have a whole lot of opinions on how those look. The opinions that it does have is that it's tab separated text. And so you can create this in Excel, or you can create this in Google Sheets, or you can just create a tab separated text file. And so you would just export from those tools as a TSV or TXT with tab separated text values. Like different tools are going to call them different things. The thing that it does care about is there's a few different names that this first column has to have. And so the first column has to be your sample IDs, and they have to be unique. Beyond that, we don't have a whole lot of opinion on what they look like in terms of characters, anything like that. We do have some recommendations in our documentation. Like, for example, we don't recommend that they're longer than, I think we say 128 characters long, mostly because there's some other tools that we work with through QIIME2 that have constraints on those sample IDs. Aside from that, you can put any other information that you want in this file to associate with these, but it's all optional. So the only column that's actually required is this sample ID column, and that makes it a sample metadata file. There's a few names for this column. It can be sample ID, it can be ID, it can be sample name, and then there's a few others. And that's just a requirement so that we know that we're looking at a sample metadata file when we open one of these up. You can also always have more samples in this file than you have in your sequence data or in your feature table. So if you're ever filtering, say, samples out of your feature table because they're replicates or something like that, you don't need to remove them from the metadata. The basic idea here is that we want to encourage you to have one master copy of your sample metadata and use that throughout. When you start having different people editing metadata files, it becomes really easy for them to diverge and Somebody fixed an error in one copy of the metadata file, but that didn't get updated in this other copy of the metadata file. And so that's where we see a lot of confusion and a lot of issues come in. And so if you're ever like working in QIIME2 and you think I need a new metadata file or I need to make a copy of my metadata file, back up. You probably don't need to do that. Reach out if you have some questions, like great stuff to ask about on the forum. Like if you're wondering, how do I manage this? That's all fair game to ask on the forum. Okay. I do have a little note down here at the bottom. This is a tool that years and years ago, one of my master's students developed, but it helps with creating metadata identifiers. QIIME doesn't care what the identifiers are, but we built this tool that essentially creates what we call like error correctable sample IDs. And so basically it will give you a set of IDs that are all multiple characters different from each other. And so it helps with like transcription errors, say when people are jotting down sample IDs from a tube in a lab. It just helps with correcting the kinds of errors that get introduced there. Okay, so typically then what we're going to have is, somebody is going to do some DNA extraction. They're going to do some DNA sequencing. This is typically going to be a multiplex sequencing reaction. And so your barcode sequence is one thing that might go in your metadata file. If you are getting demultiplex sequence data from whoever's doing your sequencing, they may already take care of that. You may never even know what those barcodes are, in which case that's also fine. And so typically, like, you're either going to get something that looks like this, like you're going to get multiplex sequences. This is a single end read example. If you have a paired end, you may have two of these sequence files, one for forward reads and one for reverse reads. This would be an example where these sequences are still multiplexed. And so you would have like 2 or 3 of these FASTQ files typically. So that might look like this. In this case, like there's going to be some barcode that's telling you which sequence is associated with which sample, and I've encoded that all with colors in here. Sequence demultiplexing, this is something that you can do with QIIME2, or you can just start with already demultiplexed data. I typically recommend that, like, if your sequencing center is giving you a choice between them demultiplexing and you demultiplexing, just have them do it, because they know what indexing scheme they use. They know what barcodes there are. They know if you should be searching for the forward barcode or the reverse complement of the barcode. Just have them do it and give you demultiplex sequences, and that will save you a bunch of headaches. And like, honestly, that's a big thing that we end up helping people with on the forum, is trying to sort this information out. If you just ask them for demultiplex reads, you can skip that whole thing. Then typically what we would be doing is getting into quality control of those sequences. And this step, it tends to be integrated with defining what we call the feature table. And so the feature table is one of the data artifacts that we were using yesterday. So in this case, you have samples on one axis, you have features on the other axis, and then you have counts of features on a per sample basis as the cells in that matrix. Now, in a typical amplicon survey, your features are going to be amplicon sequence variants. And so those are going to be unique ribosomal RNA sequences that were observed in your study. And so what this step will typically output also is what we call this featured data artifact, and that's where the actual sequences are. And so the identifiers that you're going to get, like you can see, I'm calling this feature 1, feature 2, feature 3. This is not exactly what they look like by default in QIIME2. There's some reasons for that I won't get into right now, but basically those identifiers are really not intended to be very useful for you. They're intended to be useful to like map information across different files. So different types of information about these sequences. In this case, mapping to the sequences. And so this table I mentioned is one of the artifacts that we started with yesterday. One of the other one of the other sticking points that somebody mentioned was like just a little bit of confusion still over that term artifact and what that means exactly. And so basically like what an artifact is, this is a pretty commonly used term in like data science. It basically means it's like the result of some process that you ran or that the computer ran. And so there's this sort of unfortunate additional use of the term. Like we'll think of like a sequencing artifact as like some bias that was introduced into the system. These are like, if you look up the definition of artifact in the dictionary, you'll see like there's these two different meanings. And so we're kind of using it in the sense of like, this is like an archaeological artifact, not exactly, but we're using it in that sort of use of the term. And so basically meaning it's something that was created, a result of some process that was run. Okay. So some of the other things that you would do in this stage of the workflow is you might assign annotations to those sequences. And the most common thing that you would be doing here in an Amplicon survey is doing taxonomic annotation. And so you would take those sequences, you would run them through some taxonomy classifier, and that would typically be based on a reference database. We provide classifiers for the Genome Taxonomy database, for Silva, and for Green Genes 2 through our resources site from under QIIME2.org. And so this would be like another upstream step that you're running. And so you would have these features, and then you would have these reference sequences, and then doing this taxonomic classification. This is just kind of like a silly little example here, but like here you're just getting, these are bacteria sequences. Usually like you would end up, at least getting down to like the family or the genus level. But you can see like here's that feature 1, feature 2 label showing up again. And so again, this is just how we like connect all this information, like the sequence with the taxonomy, with other information that's being generated. And so basically like what I would describe as like the upstream artifacts, and this is where we're starting with this analysis that we started on yesterday and then continuing with today and tomorrow. We have this feature table. We have our sample metadata. And so the sample metadata is something that you as the user would be creating. And then we may optionally have, just depending on what we're doing, we may have this taxonomic information and we may have this sequence information. Now, because we only have a limited time here, we're not actually running through all of these upstream steps. And so we just kind of use the jargon like upstream and downstream. And basically what that means is it's like, the way we use it, it's like before you get to the feature table, which is the QC, which is where a lot of things vary. Like if we're doing metagenome analysis, upstream of the feature table, we're probably going to be doing some different things than we are for Amplicon analysis. When we get to that feature table stage, a lot of the analyses become unified. And so that's the other reason why we select to do this part during this workshop, because you can apply a lot of the same ideas that we're going to be working with to metagenome data or proteome data or metabolome data. And so that is, basically where we are at this point when we're starting the analysis. Chloe, I'm going to go for like 5 more minutes just to, okay. Okay, so what you can start to do now when you get to this point, when you have these artifacts, is you can get into, the the interesting things that were driving you to study the microbiome in the first place. One of these that we're going to look at today, and what I'm going to start with here, is looking at community richness. This is a pretty straightforward thing to measure. And so it's a good one to just start with to see how we would use the feature table. But community richness basically refers to like the number of different taxa that you observe in an environment. And so like over here, these yellow flowers, yellow tulips, that would be a very low richness or low plant richness environment. So there's just this one species and we just see it everywhere. Relative to this flower garden over here on the right, which would be a relatively high richness environment. And so here we see many different types of species. And so here we're looking at plants, gut microbiome, we're probably looking at bacteria or fungi or archaea or all of the above. And so the simplest measure of community richness is what we call observed features. And that is just simply the count of different features that are observed in a sample. And so just based on that definition, can anybody tell me like what the observed features would be for that top sample, the first row? Not exactly. So that would be like the number of sequences that we observed for that sample. And maybe I should have flipped ahead one slide. But the idea here is we're saying like how many features, how many types of features did we observe? And so if you think of these as like species, we would just sort of say, these are the ones that we observed. And then like, so for this first sample, feature 1, 2, and 3, we observed. Features 4 and 5, we did not observe. And then we see a different set of features that were observed and not observed for that second sample. And so for each of these, we would say the observed features was 3. So yeah, very simple metric. It just basically says, count the non-zero values in this row, and that is the observed features. And so now just to sort of segue into what Chloe is going to jump into for us, I wanted to show this table, which looks a little bit more typical of, what we might see. Obviously still a toy example. But what I want to point out here is that there's something concerning about this table that we should be paying attention to. Does anybody have an idea on what this is? Exactly. Yeah. So the number of total, what we would call the total frequency, this would be the number of times each of these was observed, which in an amplicon study is going to be based on the number of times a sequence was observed, is very different, order of magnitude different for the first 2 samples versus the bottom 2 samples. This is very standard for sequencing studies. You can actually, like, if you think back to the feature table summary that we were looking at yesterday, there were some samples in there that had 700,000 sequences in them, and there were some samples that had on the order of 100 sequences in them. And so if you just to keep it simple again, if you think about a metric like this observed features metric, you would expect, or you should expect, that if you collect a lot more sequences from a sample or from an environment, that you're going to observe a lot more diversity in that environment. So all other things being equal, there is probably a correlation between the number of sequences you collect and the number of different taxa that you see there. An example that I like to give, just to sort of relate this back to the macro world, is like, imagine you are counting the number of plant species in different environments, and you go out to Let's see, how do I normally do this? Okay, so you go out to the desert. So let's say the Sonoran Desert, and you survey, 10 square, or say you survey a square kilometer, and you count the number of different plants that you see. And then you go to like a rainforest environment and you survey a square meter and you count how many plants you see. You might come to the conclusion in that case that there's more plant diversity in the desert than there is in the rainforest because you put a lot more effort into looking for plants in the desert than you did in the rainforest. And so what you really need to do is you need to normalize that sampling effort across all of your samples. And then that should allow you to normalize so if you do that, then you should expect to see something more reflective of reality. And so in a sequencing study, the corollary to that is how many sequences we collect from an environment. And this, as you know, to this date, we don't really think that there's anything that is biologically relevant that's driving these differences in sequences that we collect. There may be some extreme examples, like if it's a truly sterile site or truly sterile sample, you may observe less sequences. But in general, there's nothing really driving that difference beyond just random noise. And so we want to factor that out. Typically, the way that we would do that is by normalizing our sequencing depth or sampling depth across our samples. There have been many papers written on this topic. What's the right way to do it? What are wrong ways to do it? It still appears that the best approach is to select a sampling depth that you feel could be representative of your samples and do random sampling with or without replacement up to that depth for all the samples. And so like in this case, what we did, this is a random sampling with replacement, or sorry, without replacement to 51 sequences per sample. You can see this bottom one had 27. And so when you do this, you are not able to retain that sample in the analysis. And so this is always something that a lot of users are spending a lot of time trying to figure out, where should I set this sampling depth? And so we're going to jump in with that when Chloe comes up here in just a minute. I do want to mention, one distinction here. There's this idea of rarefying, and that's what we call that process of taking a single random sample from your feature table. That is, like this one-time process. Rarefaction, on the other hand, is the repeated running of that rarefying step. And so what you might do is you might take, rather than like one draw from that, you might do this 1,000 times, each time starting with the beginning feature table, doing your random sampling, generating the feature table. And that is really like the more robust approach here, because then you still are looking at all of the data that you've collected. You sample from that table many times. And so like you will see some of the rare taxa that are showing up there in the downstream feature tables that you're generating. But then the typical way that you would process this is that you would compute these metrics, like this observed features metric, on every one of those 1,000 tables that you generated randomly. And then you would average those together. And so we're going to do one instance of that here. But as you can probably imagine, doing this 1,000 times can take a lot longer than doing it once. And so when we get into what we call our core metrics analysis, we're going to be doing this rarefy step. When we do this alpha rarefaction plot, that's going to be a rarefaction process. So where we do this multiple times. Just recently, we released a new QIIME2 plugin that basically makes it so that you can run those same core metrics commands using rarefaction. And so they take a lot longer, but if you have a parallel computing environment, you can split those jobs up so that like you'll generate 1,000 feature tables. And then when it's computing diversity metrics on those, it'll send that out to however many processors you have. And so it'll help just speed that process up quite a bit. That is a plugin called Q2 Boots. The name comes from bootstrapping, which is sort of an alternative, but very closely related approach to rarefaction. The last thing I'm going to mention up here before I turn it over is after Chloe talks about alpha diversity, she's going to be talking about beta diversity. And so beta diversity is a comparison between samples. And so like, for example, a beta diversity analysis might look at all the species of plants in this garden, all the species of plants in this garden, and then compute their dissimilarity or their similarity, depending on the type of metric, based on what plants are showing up and potentially like what abundances those plants are showing up in. And so when we think of doing like ordination analysis on microbiome samples, that's going to be like downstream of beta diversity. I put a bunch more slides in on this topic toward the end of this slide deck. And so if you have this up from yesterday still, just go ahead and refresh and you'll see we have this extra slides section down at the bottom. And I go into a lot more detail on the different metrics in there. If you want to learn more about like what metrics are used, different types of metrics, you can go ahead and go through those. And then tomorrow, feel free to like ask us questions about that. Okay, so I'm going to turn it over to Chloe now, and she is going to take us back into that container QIIME2 environment, and we're going to run some QIIME2 commands. Right.
Awesome, thanks, Greg. Here, give me 1 second to kind of reorient myself on this computer. Here is our here is our terminal that we're working in, and then here is the tutorial. That's really what I'm going to be based off of for the rest of this little section. So having those two up should be perfect. And I'm going to navigate us to exploring the data with Galaxy. So I know we already moved past our Galaxy part and are fully in our Docker section, but we're going to use this to get our artifacts into QIIME2, or not QIIME2, but our Docker container. So the first thing I'm going to do is click on this drop down that says Galaxy, and I'm going to switch this to command line or Q2 CLI. And that should make it so that all of the commands are now not in Galaxy, but in CLI. Does everyone have that in their tutorial? Perfect. Okay, so we are going to do some of these steps again in the Docker container, but for the sample metadata, I'm just going to run this wget command. So I'm going to go over here and copy it, and then go back to my terminal, and I don't know where I am. Let's figure out. Okay. So I'm going to CD into the 2024 workshop directory, ls. And that way I just don't clutter up that home directory too much. And now I'm going to copy and paste that wget command. And then this is just kind of a thing that I always do, but after I run every command to get anything, I always ls to make sure that it ends up there. So I like to just kind of prove to myself that things went right. So there we go. We now have our very first kind of piece of data in our Docker container. Um, so like I said, we were able to look at our metadata tabulate in Galaxy. Oh, we copied this command from right here. So if you have this TSV, this tab separated values file in your Docker container right now, we're going to skip this metadata tabulate visualization. Visualizations in QIIME2 tend to be kind of like an output. You don't really input them into anything else. And so that's a way of viewing our metadata, but it isn't really going to be a way that we interact with our metadata. So I'm going to skip this command because we're just going to use that TSV whenever we need that metadata. But I am going to come down here to access and summarize the feature table. And I'm going to copy this wget command for our feature table. Copy? Yeah. I believe as long as it's like you can set it up so that it has like access to a bigger server or using some sort of cloud. And in that case, you would have more space. But yeah, it is limited to how much space you like allow Docker to have. That I'm not sure about. So but yeah, it can't take more resources than your own computer. However, you can run that. Yeah. Other places you have. Okay. Perfect. Awesome. So I'm going to go copy this feature table and paste this command again. wget, we're calling it a feature table.qza, and I'm downloading it from our tutorial. So after that, I'm going to ls. And I have my feature table, which is my input data for the rest of this tutorial that we're going to do, and my sample metadata.tsv. When you guys ls in your terminal right now, raise your hand if you do not see both of these things, and we'll have someone come around and help you. Perfect. All right, so after that, we are going to run this same command that you ran in Galaxy in the CLI in your Docker container. So we're going to summarize this feature table. Why we're doing this is, again, a little bit is to show you kind of the interoperability that we can use this over multiple UIs, however you feel comfortable, but also because there's some important information that will help us select our sampling depth. And so we need this visualization in our Docker build. And so we're going to copy and paste. So in this command, we've run it previously, and hopefully it doesn't take as long as the Galaxy to version of it to run. It shouldn't. And it didn't. But so we have our input table, which is our feature table, and then our metadata. And then as an output, we're getting that visualization. No, I'm not sure. Greg, can I run QIIME Tools View in here? No, I need to download it. Okay. The possible proof. Okay, so I'm downloading it onto my local computer. I'm allowing it to download onto my local computer. And then I'm going to navigate a little bit in my way. I'm going to now navigate to QIIME I'm going to Google it, QIIME2 view. And the web address is view.qiime2.org. And so this is the same exact place that you were brought to when you clicked the view with QIIME2 button in Galaxy, but we just navigated there slightly differently. And then I'm going to go to my recently downloaded right here and use the drag and drop. No, I'm not going to do that. I'm going to try again because I think that may have just been a stochastic. Oh, okay. Okay, well, Firefox allows me to do that, so Firefox is better. But so now we have this feature table summarized, and it really shouldn't look any different from what you saw yesterday. The only difference is actually this is a newer version of QIIME on our Docker installation. It's like the most recent one compared to Galaxy, which I think was 22.11. And so if we went to the provenance, we could see that this summarize command was ran in a different version of QIIME2. Oh, God. Right there. You can see the version of QIIME2 is 24.5. So the most recent you got. All right. So now I'm going to navigate us to our interactive sample details page. Let me know if you aren't able to pull up this interactive sample detail page. All right. So I tend to navigate towards like a couple of metadata categories that I think are important. I feel like most people who are analyzing their data have a couple of metadata categories that you really want to understand. One for me would be patient ID. I want to make sure that we have samples from all subjects. And then another one that's really important is time points. For us, which looks at like, this is hard to read. I can zoom in a little bit. Oh, that's bad. But this looks at like our consistent time points through the FMT. And another one of interest is this treatment, auto FMT group, which allows us to tell whether or not it's a treatment or a control sample. Now, when we're dealing with sampling depth, this slider kind of gives us an idea of what we're going to lose and retain as we choose a sampling depth. And so if I slide this all the way over, I keep one whole sample, which is super exciting, but I lose all my other samples in the process. But the positive of this is that I can investigate that sample as much as I want. The negative of it is that no statistical methods allow you to run on one sample. So I don't think that will be my sampling depth. But here we're kind of playing a balancing game where you're kind of playing a balancing game where you either are going to lose samples because they don't have enough sequences to meet your minimum depth, or you're going to throw away features because you have sequenced more features and samples, but you don't have enough across your samples to look at all of them. So a good example would be this first one. If I set the sampling depth at 10,000, even though we have 700,000 sequences in this sample, we will only be looking at 10,000 of those 700,000 sequences. And so it's really important to just understand like kind of the range of your samples and kind of what is important to maintain. I think, yeah. Yeah, it's kind of hard. So in this specific study, it kind of depends on what you're favoring. I tend to favor features, meaning I'd like to investigate the sample as much as I can. And so I'm kind of willing to throw out some samples that have lower sequencing depths in order to make that happen. That's only super possible when you have like replicates within a group, right? Like I'm willing to lose some of these controls because I know I have more. With a study like this, it becomes hard because you have time points. And so if you start throwing out samples, you could lose a subject at that time point and you don't have any replicates of that. And so that tends to be where I kind of switch my favoritism and I start going towards, I want to retain samples and I'm okay investigating a little less. And so I think it is just kind of a balance that you have to figure out per study. And in this case, I am going to kind of prioritize us keeping samples so that we're able to investigate a subject throughout time more effectively. Yeah. Does that answer your question? It was kind of an it depends answer, which is kind of the answer of the workshop. But another thing you have to look at. So I'm going to leave this page a little bit, and then we're going to run another command that's going to help us kind of decide our sampling depth. So if you go back to our tutorial page, and you go to selecting an even sampling depth, we have this alpha rarefaction plot. And what this does is it allows us to see kind of when our alpha diversity levels out. And that allows us to kind of see if we've chosen a representative sampling depth. And so I think this is best explained if we just run it and take a look at this plot. So I'm going to copy this right here. So yeah, selecting even sampling depth and then navigating to the bottom of alpha rarefaction plots. And then I'm going to navigate back to our terminal. And I'm going to paste that alpha rarefaction command. I don't understand. I just like double paste. But all right, there we go. And so now we're running this alpha rarefaction plot. Raise your hand if you are not able to get this running. This might take us a little bit of a second. And so people can come around and help if you're having issues getting this to run. Yeah. Oh, yeah, that's a great question. Thank you for bringing that up. So with alpha rarefaction, we kind of choose a max depth that we're going to start testing for. So I haven't necessarily chosen 33,000 as my sampling depth, but it's kind of like where I'm going to test out to like see how sampling depths look out to 30,000 or 33,000. This is like important. I tend to choose a couple of max depths if I'm playing around with it, because if you choose a shallower max depth, you get more like a better idea of the points in between it, because I think it samples at 10 random points. And so the larger it is, the kind of bigger spaces between your max depth. And so I kind of tend to play around with it, but this should give us like a general look at it. Some people recommend using the median of your data's like max depth or as your max depth for this, but I like to just kind of play around and see what the data says. Mm-hmm. Yeah, I think it's something here. So I'm just pulling up another terminal so I can run this help. Shine. I didn't run help, so it's going to yell at me, but it will give you the docs. But basically you can choose the amount of iterations that you want to do in between. Yeah, using this P iteration. No, sorry, that's not right. P steps allows you to choose how many steps happen in between your min and max step. And then you can input your minimum depth, but that's also defaulted at 1, so I tend to not. And then you can also choose how many iterations of that rarefaction that you do at each step. Yeah, thank you. So I'm still waiting on this alpha rarefaction to run. This step kind of takes a while because we are like subsampling things a lot of times. So because it is, rarefaction where you're doing this a number of times, we are subsampling it multiple times, and then we're doing that at multiple steps of depths. So we end up taking a while. Okay, mine ended up running. I'm not gonna wait until everyone's runs. I'm just gonna steam ahead and show you the results. But so here on the side on my file finder, I can see I have my observed features alpha. I guess the name was a little bit too long for this GUI, but we're gonna download that again. And I'm gonna pull up another view.QIIME2.org tab. And I'm gonna drag and drop again the observed feature alpha rarefaction. So this is coloring by patient ID right now. You can change this depending, or drawing lines based on it right now. But we can see kind of that, I would say that like between 3,000 and 6,000, there's not really lines on that, but we can see that this range right here, it's still kind of climbing in alpha diversity. And it generally is still climbing until we get around maybe 10,000, 12,000-ish. And then it kind of starts to level off, although that's not the case for all of them. As you can see, this guy right here, as you go deeper in the sampling depth, he's consistently climbing in alpha diversity. And so I'm not sure that we're ever really seeing like a level off for that specific patient ID. But we're also kind of looking at general trends here. So we can see that this is generally leveling off around 10-ish thousand. Oh yeah. And so the lower plot here is looking at sampling depth and number of samples. And so it visually kind of tells you, as you're choosing a sampling depth, how many samples you're going to lose from whatever grouping you have. And so we have patient ID, but here we can see that like as we go down in our as we go up in our sampling depth, we get a deeper sampling depth. We are starting to lose samples from those patient IDs, which again is just kind of part of the trade of creating an even sampling depth. But in the case of a longitudinal study, we really want to prioritize keeping those samples from all of our patients around. All right. So I'm going to go, Yeah, I typically prioritize kind of both. I feel like this information, this plot specifically that we're looking at right now, I also kind of learned from this information by kind of sliding this and seeing how many plots or how many samples I lose there. And so I consider all of this information and how the plots are evening out. We want to make sure that like I have an amount of sequences that I feel okay investigating. I have an amount of samples that allows me to make statistical analysis that I need to, and also that I'm choosing a reasonable sampling depth. Because if I choose one that seems to not be at a stable place in alpha diversity or in other diversity metrics, I probably am not really representing those samples very well. And so, just a balance of representing the samples as best we can without losing too many features or too many samples. Yeah.
Yeah, one of the other things that I look at when like with this plot is I use it so like see this blue line is looking kind of odd, right? Like it seems to have leveled off and then all of a sudden it just shoots up. So why might that be? And if we were to look over here, it looks like I'm thinking that this is the same line. And like what happens is like at that point, it looks like we lose a bunch of samples from that subject. And so that suggests to me that like where maybe before we're averaging across 25 samples Now we're getting down to averaging across fewer samples. And so that can explain like why you would get that big jump at that point. And so that's why I like to look at this one too, is just to try and interpret like why might things be changing in that upper plot. This can also happen if like your lines look really, really stable in there. Like if there was only one sample for one of these groups, it would, potentially look like, it was flat because you are just looking at one sample at different depths. And yeah, it's just important to know like how much, it's essentially like putting an N on each of these boxes here. Yeah. Yep.
Awesome. So I'm gonna so I guess we should decide a sampling depth now, is kind of the large point. And so here, I told you guys that I'm gonna prioritize keeping samples because it's a longitudinal study and I want to see everyone's trend over time. And in alpha rarefaction, I told you guys that I'm looking for a place where this feels like it evens out and all of the samples look reasonable in order for me to get a reasonable sampling depth. So I just wanted to confirm, I switched over just to confirm that I don't choose the wrong sampling depth for this tutorial. But so 38,000 is a little too much for me. In this tutorial, we choose 10,000. 10,000 means, so I'm looking through all of our samples. 10,000 sampling depth means that we keep a lot of samples. As you can see, this study has a ton. And we're kind of keeping most of them. I also tend to look for a jump in our samples here, just because it makes it like an easy cutoff place. Like if I can see that there's like a jump from like 9,000 up to 12,000 and like a 3,000 difference between two samples, I tend to look for that. We didn't do this in this tutorial, but I like to max min my sampling depths. And so I tend to choose like exactly the minimum sample that I can. So I would probably, if this was my own analysis, choose 10,112 because that's like, I can investigate all of the features in this sample, and then I get 112 extra samples to investigate with every other sample. And so that's a little tip that I do. But with this one, we just chose an even 10,000 here. And so that's what we'll be doing. So switching back over to the workshop, we're going to be running our core metrics command. And what this does is it runs a bunch of our like very popular metrics that we're running. We're running this non-phylogenetically. And so we're going to stay away from the unifrac and like the unifrac and faith PD stuff, but we're going to get observed features, Shannon's, evenness, Jacquard's, and Bray-Curtis from this. And as Greg said, so most of these are explained in those extra slides that are at the bottom of our presentation if you guys are interested in more information. But so we're going to run this and it's just going to kind of give us a huge amount of different diversity metrics that we're going to explore, continuing on tomorrow when we look at Q2FMT. And the last thing I'd say is that we also get this rarefied table here, which is what we are going to use in order to run all of these diversity metrics. That's the table that's going to be rarefied, just like Greg's example showed, down to whatever sampling depth you showed, so that every sample has a total frequency of 10,000. So now we're all on the tutorial and we're going to copy and paste QIIME diversity core metrics. And we're going to paste that into our working directory. It doesn't like when I command V. I keep trying and it's done that to me twice now. I don't know. It's like a paste, but it's fine. Okay. And so we're going to run this and get a bunch of core metrics. I don't think we have time to explore these visualizations today. So I might, do you want me to check out a Ray Curtis one or Jacquard? It should take that long to run, and then I'll look at it. Okay. So we're going to briefly look at one of the emperor plots, but we're running short on time, so that will be kind of quick. But yeah, so we'll generally look at these core metrics that are generated from this plot. Oh, and there it is. This is something we consider a pipeline in QIIME2. So there's a lot of actions being run under the hood that make this just one command, which is kind of awesome because I wouldn't like to run all of them. All right, and so then we are ending up with this new diversity core metrics directory. And when I peek inside of it, I can see that it kind of lines up with all of this green text that I'm seeing. And so I'm going to download our Jacquard Emperor plot. I'm prioritizing Jacquard because we're going to look at that with using Q2FMT tomorrow. So familiar. It will be familiar tomorrow. Right now it's not. And I'm going to exit out of here. And I'm going to drag and drop this Jacquard Emperor plot. So how many of you guys are familiar with Emperor plots? Okay. So Emperor plots are a representation of beta diversity. So like Greg said, it's kind of a representation of how similar or dissimilar two samples are when you're comparing them to each other. And so an Emperor plot represents a visual distance between those two samples. And so the TLDR of an Emperor plot is that when I look at two samples and they're really close together, I can tell that they have a pretty high similarity of features. But when I look at this sample, which is FMT92B, and this sample, 92L, that was just, so those are actually from the same subject, but that was just random chance. If I look at those two samples, those are really far apart. And that indicates to me that those samples are not very similar. So it's just a 3D representation of how similar or dissimilar samples are based on how close they are together in the plot. So looking at this plot, would you say that these samples are similar or dissimilar? Yeah, they must have a pretty similar beta diversity to each other because they look really similar. And they aren't the same subject at all. So that's just two people that have the same similar gut microbiome. But this doesn't exactly tell us how similar they are. They're just pretty similar to each other relative to all of the rest of the data, which is important to note. Something you can do with PCOAs is you can decide to, hide a group. But I would say that that isn't like a super representative thing to do because this is a 3D plot of all of the distances. When you hide half of your data or some of your data, you're kind of hiding the distance relative to other points. Like if I just plotted this data by itself, it may look different because treatment and all of the samples that are related to treatment play a big role in where those dots ended up being plotted in this PCOA. And so that's all just to mention that this is a pretty relative plot that changes based on what samples you include in your distance matrix. I am going to look at this really quickly. What we like to see is clustering. That's what people typically point out in their papers is like, oh, we see our treatment and control group, they cluster distinctly or they don't. Here, when I'm looking at treatment and control, surprisingly, I don't see really any distinct clustering. And I kind of have a hypothesis for why that might be. Does anyone else have a hypothesis for why treatment and control don't seem to be clustering distinctly? So I think it's because it's like longitudinal data. This isn't really capturing the changes of a specific subject over time. It's just plotting them all together. And so I think if we were taking into account like how the sample was changing from the last sample it had, we would have a better picture of how this data like looks longitudinally and how it's shifting and what those differences are. But without taking into account that really core variable of time, change over time, we're not really capturing anything. And so it looks like our signal is really weak. And I guess that's a perfect lead in for our next section, which is looking at this data, but using our plugin Q2 longitudinal. Q2 longitudinal, as the name implies, creates longitudinal data or analyzes longitudinal data and allows us to really interpret that.
All right. Thank you, Chloe. Okay, so yeah, so like what we just did here was ran a couple of different QIIME2 analyses. These are kind of like classic plots that people are familiar with from QIIME2. And like I said earlier, almost always like some of the first things that I will do when looking at data from a microbiome study. At this point, usually your analyses tend to become a little bit more customized to the specific questions that you're interested in. And so, like where Chloe left off, she said, we looked at this plot. I would look at this plot and I would say, okay, things generally seem to be looking pretty sane here. Like if I go back to the patient ID column, it can be a little bit hard to see in here because there's so many. But like I see, for example, like these gray samples, which are going to be one particular patient, this patient FMT 0063. They tend to be like grouping closer to each other relative to other subjects in here. And like in general, like if I look at these purple samples up here, those are going to be from one subject again, just because of how I color this. So like I would kind of look at this and be like, sanity check. Yeah, this looks sane. And so now what we're going to have everybody do, and since we're basically at the end of the day here today, I'm just going to quickly point you at this. And I want you to do this at home or back in your office this afternoon or tomorrow morning. But we're going to have you jump into a more specifically focused analysis. And that one is going to be the next one that is in this section. And so we have a link to a paper in here on a QIIME2 plugin called Q2 Longitudinal that was developed, like Chloe said, for longitudinal microbiome data analysis. And we have a command in here that does linear mixed effects models on this. And so this is going to be an analysis that is taking into account the fact that we have repeat samples from an individual subject. And so what I want you to do tonight is run this command, look at the results, refer to that paper if you need to get some help with interpreting it. And basically take a look at like whether you're seeing an association in our dependent variable. So in this case, that's going to be that observed features metric that we're looking at. See, is there an association with time? And we'll include the treatment group in there. And so does treatment impact whether you see a association with time? And so this is like a pretty general purpose longitudinal microbiome analysis approach. And then tomorrow, what we're going to do is we're going to dive into something that's even more specific for this type of study, and that's with the Q2 FMT plugin. And we're going to ask some very specific questions at that point. So we're going to ask questions like, did the recipient's microbiome get more similar to the donated microbiome following treatment? And so we'll approach some of those types of questions tomorrow. So we need to transition now to just to sort of wrap up for the day. We'll be around a little bit if you have some questions. And then I just wanted to remind everybody that at the end of the day tomorrow so like our program will officially end at 2 tomorrow, but we're going to stick around for an extra hour after that for optional Q&A. And so if you go home, poke through some of these slides, poke through some of the results, have questions that either we haven't gotten to yet or that we don't get to tomorrow, we're around. We can also use that time. Like if you have something like my metadata file is not loading with QIIME2, we've got some folks around. We can like sit down and look at your computer with you and, try and poke around with it tomorrow. Okay, so with that, I'm going to turn it back over to Carrie, who's going to wrap us up for the day.
I just have to pull up. All right,